presentation from our community engagement core. So want to thank everyone for joining this new date today. So quickly, my disclosures. Today, I'm going to talk about an experience, a ongoing experience that I've had with the VA Rheumatoid Arthritis Consortium. And I want to talk about how this consortium and really team science that we've conducted as a group has allowed for rigorous clinical translational research, but also has served as a very important platform for career development and helping junior faculty and trainees, even pre-faculty, get important experiences in clinical translational research. So I'm going to talk a lot today about the VA. That's really a backdrop. I am a professor here at UNMC, involved in a leadership level in the Department of Medicine, but I am all in when it comes to the VA. And I think often when people think about the VA, they think about, the cartoons, the political cartoons like this that they see. And I think sometimes as researchers, we change that a bit. And we feel like this, with the IRB and the regulatory requirements of doing research at the VA. But this is really an inaccurate picture. And I hope that you get that feeling and that message comes across today as I talk about our efforts, which I think are similar to other experiences people have had in working in the VA environment as part of team-based interdisciplinary research. So I'm going to talk again about the VA system, and just as a little bit more background, the VA is by far represents the largest integrated healthcare system in the United States. There are over 1,400 VA hospitals, over 15,000 healthcare providers working within the confines of the VA. Each year, the VA provides care for approximately 5 to 6 million US veterans. The VA is involved in many of our clinical training. In fact, a majority of US providers have had some training in the VA system. I think it's about 2/3 of providers in the United States received at least part of their training in the VA. Important to this talk, the VA really and truly has been a pioneer in the development of the electronic health record system. And that's important to this talk because we have been able to leverage those early efforts in data collection, et cetera, into our registry efforts. And I'll talk more about that as I go. So a little bit more about the VA. I mean, we think of the cartoons I talked about earlier. I don't think we necessarily immediately think about research when we hear about the VA, but the VA has done a tremendous amount of research historically. And if you look at some of the groundbreaking things that have come from the VA, this includes Nobel Prizes in science, but includes really important things like the first liver transplant, development of the artificial pancreas in terms of transplant medicine. It includes the development of anti-malarial therapies, some of which we have stolen in rheumatic disease care, and trials in tuberculosis in the 1940s that really pioneered treatments for these infectious diseases. So the VA has made major contributions in healthcare discovery and moving healthcare forward in the United States since its early inception. And I'll just point at the bottom here, I have some representative veterans shown, and I'll just say, as a really important side note, that the patients in the VA health system, so US veterans, are just amazing in terms of research. So I think from a personal experience, veterans often see this as a way of extending their service by participating in research. And it's been a tremendously gracious and willing population to do research with. So in addition to the other highlights, I'd be remiss if I didn't also show highlights that have come from our rheumatology group in the VA system. So this is work we published in the New England Journal in 2013, looking at different therapies in the treatment of rheumatoid arthritis, which I'm going to talk about today. We have another cooperative study that we recently directed in gout that we hope to have a similar publication for in the upcoming months. So really, really fantastic efforts, both led by Dr. Jim Odell, who is the chief of our rheumatology group, both here and at our Omaha VA. So just a little background, because I'm going to talk a lot about rheumatoid arthritis today. Rheumatoid arthritis is often said to be the most common form of autoimmune inflammatory arthritis worldwide, affecting probably up to or a little below 1% of the population, much more common in women. And important to our talk today, this is a systemic disease. This isn't just a disease of joints. It is a full body problem. So extra articular involvement with the involvement of lung tissues, heart tissues, eyes, skin are all too common in our patients, and particularly common among men and patients who are smokers. It is a female predominant disease. It is a very costly disease. And so the cost of taking care of a rheumatoid arthritis patient over their lifetime is now more expensive than taking care of a diabetic. And this has largely been driven by the expensive nature of novel therapies that have evolved in the treatment of rheumatoid arthritis over the last few decades. Rheumatoid arthritis is a classic chronic condition, and it has both genetic and environmental risk factors, and really is noted for a reduced survival and increased disability. So major indirect costs in rheumatoid arthritis. So this is a typical timeline that people teach medical students about in rheumatoid arthritis. You have early disease, and I think everyone here can appreciate the swelling of these small joints of the hand, often a symmetrical arthritis. As this advances with suboptimal treatment approaches, this leads to progressive deformities in the hand, and you're starting to see in this middle picture at 5 years, subluxation of joints. You're starting to see what's called ulnar drift. And these are hands that are increasingly getting more and more difficult to do activities of daily living with. And then by 10 years or longer, in some untreated or suboptimally treated patients, you can see really just devastating forms of arthritis. Now, just as a sidebar, this last picture was a patient of mine, actually, who recently passed away. He was a medic in the Korean War theater. And interestingly, when I quiz students about which set of hands they think own these 3 hole-in-ones shown in this news clipping, it's the hands on the right. And so amazing resilience in these patients. And as a provider for these patients, it's been really inspiring to be able to work with these patients in the VA system. So I came to UNMC after my training in 2002, and during my training had experience with cohorts, including the Iowa Women's Health Study and the CLEAR Registry, which were studies, longitudinal cohort studies of patients with rheumatoid arthritis. And part of my job was to take on rheumatoid arthritis care at our VA health system. And so the thought was, at the time, just really extending my prior work, is this something that we could do in the VA health system? And we had a RAIN network at the time, which served as a model for collaborations and investigator-initiated research. And so that was the big question we had in 2002, is really, could we do this in the VA? So we also knew at the time there was already data emerging about differences based on biologic sex in terms of RA disease severity and phenotype. There was data from the Mayo Clinic. This is the famous Olmsted County Registry, which I think has had about 1,000 papers published from it in the rheumatoid arthritis alone, but showing, back in the late 1990s, that there appeared to be some differences between men and women who had rheumatoid arthritis, namely that men had more erosive disease. So really that's a way of saying more destructive arthritis. They had earlier onset disease, but perhaps had a slightly less functional impairment related to RA than women who had RA. Really importantly, even from this early data, it appeared that men had much higher rates of extra articular disease, and in particular had much higher risk of having interstitial lung disease, which is a big problem because the mortality related to interstitial lung disease in the context of rheumatoid arthritis is quite devastating, with some papers publishing median survival rates in these patients as low as 3 to 5 years. Men also appear to be more likely to develop subcutaneous nodules, which can be very problematic. And men were more likely to have autoantibody with their disease and have higher concentrations of autoantibody, which is important because these autoantibodies predictably associate with worse disease outcomes. So back in 2002, there were several ongoing cohorts that were focused in rheumatoid arthritis. I mentioned the Iowa Women's Health Study. I mentioned the CLEAR Registry, but there are these others, the Women's Health Initiative, the EIRA, which was out of Sweden, the North American Rheumatoid Arthritis Consortium, and the Nurses Health Study, which is still ongoing, led by researchers out of Brigham and Women's in Boston. And not surprisingly, based on the demographics of rheumatoid arthritis, but also you can see from the names of some of these studies, these cohorts were either a majority women or completely women and excluded men altogether. And so there was this opportunity in the VA to perhaps learn about rheumatoid arthritis in this very understudied, and you might argue unstudied, subset of rheumatoid arthritis patients, that is men, older men with rheumatoid arthritis. So about this time in 2002 was also when new therapies, sort of the often called the biologic era, was emerging in the treatment of rheumatoid arthritis. So you all watch enough TV that you've seen these commercials for drugs like Enbrel, Remicade, the newest and greatest treatment, Humira, which is one of the leading prescriptions in the world. These are therapies that cost out-of-pocket expenditures of about $80,000 a year. So this has become a very, very expensive treatment. And rheumatoid arthritis has become a very important disease, not only for patients and rheumatologists, but now also healthcare systems. I mentioned earlier as well that the VA really pioneered the development of the electronic health record. And here's a front page or a snapshot taken of the CPRS electronic health record system that's used in the VA. This actually rolled out in the VA at the time I was a fellow, and that tells you how old it is. So this has been around for more than 20 years and is finally currently undergoing an upgrade with the transition to Cerner systems across the nation. So it was in that context that we developed this registry called the VA Rheumatoid Arthritis Registry in late 2002. And our goal at the time, which really hasn't changed, that we put on paper, was to establish and maintain a national, clinical, and a biologic repository that would facilitate investigator-initiated research, that would improve our understanding of RA-related comorbid conditions, and I'll come back to that. It's a big deal in rheumatoid arthritis. That would lead to interdisciplinary collaborations that would ultimately improve outcomes for our veteran patients with rheumatoid arthritis. And I'm going to come back to this, but we really wanted to develop something that would provide a training platform for trainees interested in rheumatoid arthritis research. And this is a picture of our very first investigator meeting held in Omaha in 2005. So 3 years into its development, we had our first face-to-face meeting in Omaha. And there's some really important people in here that helped lead early efforts from other VAs around the country, including Stuart Richards, who is at the Washington, D.C. VA, Gail Carr, also from D.C., Dino Kazi from Dallas VA, and Grant Cannon from the Salt Lake City VA. So this was a map of our early efforts in 2004 with these 4 sites, geographically spread, but pretty limited. And now this is what VARA looks like several years later. We now have 16 active sites across the country. These are all VA sites. And I think what's impressive about this is you have to remember that we offer no direct funding for site participation. That is $0. And so these are sites that have joined our effort and have contributed in very meaningful ways in the absence of direct funding. And so that tells you a lot about the interest of rheumatologists and rheumatology clinics in improving healthcare for our patients. So when we started the registry in early 2003, so the first patient was enrolled in the VARA registry at the Omaha VA in my clinic, with me serving as the study coordinator in January of 2003. And this is what our database looked like at the time. We got informed consent on patients. We collected baseline data clinically with some very simple streamlined forms. And then longitudinally, we collected disease activity measures in rheumatoid arthritis as part of routine care. So there were no scheduled study visits. Patients came in when they came in to get care, and we got permission from them early on to collect that data over time. And we had a programmer, actually a collaborator who wasn't involved in VARA, but who knew how to do access programming, developed this relational database for us. And this is how we started out, very rudimentary efforts in early 2003. A key to our efforts was that all the veterans gave us complete access to their medical records moving forward indefinitely, which was very wise move at the time. So one of the things I want to point out about these efforts is these kind of efforts, these multi-center cohorts, take a long time to mature. So this is the very first paper we published on VARA in 2007, a full 5 years after we had initial discussions about the registry. And it was a very simple descriptive paper, essentially looking at race, ethnicity differences in disease activity and severity. And what we showed essentially was there were very little differences in groups based on race, ethnicity, with a few minor exceptions, particularly in terms of the frequency of subcutaneous nodules, which seem to be a little less frequent in Black or African American patients with rheumatoid arthritis. So obviously, like all good research efforts, longitudinal efforts, our effort has evolved over time. And so the VARA registry, I showed you kind of the simplistic database. We now have evolved to a place where what we do is all of our data goes into routine clinical notes, and those clinical notes are automatically abstracted using a process called natural language processing, or NLP, to automatically pull the data we need from notes, requiring no data entry, no data collection efforts on the part of enrollment sites. So you can imagine how easy, or how much easier, this has made it on our site personnel. And so to date, we have enrolled over 3,000 patients into the registry. We have over 30,000 clinical observations and growing. And we have an average follow-up of patients of at least 4 years. It's getting closer, I believe, to 5 years now. So at the same time we were developing this in 2002, I was involved in early training efforts and committees in the American College of Rheumatology. And as part of one of those early meetings, I had the fortune to interact with a gentleman named Peter Gregerson, who has had major contributions in our understanding of rheumatoid arthritis. Namely, Peter's work is responsible for identifying the major genetic risk factor for the development of rheumatoid arthritis, that is HLA DR beta 1 alleles, or the shared epitope allele. And Peter was giving a talk to this young group of scientists, and he made this comment. He said, if you develop a bioassay, you're a beggar, but if you develop a biorepository, you are the king. And when you think about that, would you rather be a beggar or would you rather be a king? I think the answer is pretty easy, and that informed really our thoughts and our efforts early on to tie the registry to a biobank. And so we did just that. So we developed a very simplistic biobank that essentially collected samples at the time patients enrolled into the registry. So when people enrolled in the registry, and this was all patients with rheumatoid arthritis, we banked serum, we banked plasma, and we banked genomic DNA on these patients. As we got these samples in, we did standardized laboratories that are meaningful in rheumatoid arthritis. So we did autoantibody measurements, anti-CCP and rheumatoid factor, which are clinical measures that we use in the clinical setting all the time. We did these in a standardized fashion. We did C-reactive protein levels, a measure of inflammation, and we looked at the major genetic risk factor in all these patients, this HLA-DRB1 allele that I mentioned a second ago. And so these variables are available on essentially the entire VARA cohort, and we update these not in real time, but in near real time in batches as these specimens continue to come in. Over time with research, the number of biomarkers that we've obtained has grown. And these biomarkers, by agreement with people who lead these efforts, become part of the VARA registry. And so we now have multiplex cytokines and chemokines available on over 2,700 of the VARA registrants. We have novel autoantibodies, vitamin D concentrations, whole genome SNP data. We have adipokines measured, and we have others. And there will be more to follow. And so that I am confident of. So we now, with this, have the VARA registry data linked with biobank data and available biospecimens as people want to measure and evaluate new biomarkers for disease. So just this linkage early on allowed us to participate in some important national efforts, and in discussions with Peter Gregerson, who I showed a picture of a second ago, we were able to join international consortia early on that were chasing down non-HLA genetic risk factors in rheumatoid arthritis. And you can see here, this is a Manhattan plot looking at different chromosomes and risks for rheumatoid arthritis. So you can see the HLA gene I talked about on chromosome 6 here, sky high, skyscraper, tremendous risk factor for rheumatoid arthritis. But you can see all these other non-HLA risk factors that popped out. And VARA was able to contribute to this effort early on, just with the simple biobanking effort. So we've subsequently been able to examine other important questions, questions that really loom over the rheumatoid arthritis world. And, is rheumatoid arthritis associated with survival? And the answer is a resounding yes, unfortunately. And so in early work, we showed that having rheumatoid arthritis, if you're a US veteran with rheumatoid arthritis, compared to age-matched men in the general population, you are at a much higher risk for mortality, more than twofold risk for early death because of your rheumatoid arthritis. And this is a figure just showing associations of other factors with mortality, including BMI. And we found this really interesting phenomenon where there was this U-shaped association with mortality. And indeed, we saw that underweight individuals, so patients with BMIs under 20, were at the greatest risk for mortality. And that was not what we necessarily expected, but definitely what we saw. And that's been a consistent finding, not only in our database, but in others that have followed. You can see associations, dose-related associations of inflammation with mortality in rheumatoid arthritis. And you can see our #1 treatment in rheumatoid arthritis, methotrexate, has a protective effect with all-cause mortality, while prednisone is associated with higher mortality. And again, those are observations that have been replicated by others. We were able to do early work trying to decipher what the heck is rheumatoid factor, which is a measure we've used in the clinic since the 1950s and the 1960s. What does it mean? This has been a long-standing question for rheumatology researchers. And we worked with Jeremy Sokolov, who was at Stanford University at the time. And Jeremy had this really interesting hypothesis that rheumatoid factor was really an amplifier of inflammation. And specifically that it would potentiate the effects of this autoantibody. This stands for anti-citrulline protein antibody. This is an autoantibody that is very specific to rheumatoid arthritis. And that these two would act in concert to amplify inflammation. And so he did work not only using VARA samples, but also did this clever in vitro work to look at this hypothesis. On the left, these 6 bar graphs, you're actually seeing inflammatory markers in patients from VARA based on their autoantibody status. So going left to right, you're seeing patients who are negative for both of the autoantibodies, one autoantibody alone in the middle 2 bars, and then the bar on the right, you're seeing patients who are dual positive for both anti-CCP or ACPA and rheumatoid factor. And what you see is that these do seem to amplify each other in that those who have both autoantibodies have the highest burden of inflammation on these cross-sectional blood samples. Jeremy then took that question to the lab and stimulated macrophages with different, with rheumatoid factor alone, with ACPA alone, or immune complexes that included both autoantibodies. And universally in these studies, he saw that when you exposed these macrophages to both autoantibodies, immune complexes from these autoantibodies, it amplified the immune reaction from these cells. And so this was a very, I think at the time, very novel concept that we published in 2014. So I think I always describe this as kind of an aha moment for VARA, or a watershed moment, was work that was led by a colleague of mine at the Salt Lake City VA, Grant Cannon, who's recently retired, and one of our other collaborators, Brian Sauer, who's a bioinformatician at Salt Lake City and the University of Utah. And what Grant did is he did this early effort where he made attempts, and he saw the value of this early on, as he attempted to merge our VARA registry with national VA data. And Grant understood even in 2011, 2012, that that was powerful, because if we could successfully link VARA registry data with national data, there's really no end to the types of scientific questions that we could ask. And so Grant performed this very simple data where simple concept, at least, not a simple study, but a simple concept, asking the question, if we look at adherence to rheumatoid arthritis therapies using national pharmacy dispensing data that's available on everyone in the VA, could we predict disease activity using the VARA registry? And what he showed in the study is that we could, that patients who were less adherent to their therapy had higher disease activity. And while that alone isn't necessarily a novel observation, the journal, this was the Arthritis Care and Research, which is one of our major journals, understood the significance of the methods, really, and the importance of being able to link VARA with national data. And indeed, the number one bullet that was published under the significance and innovation section of the paper was, clinical data in the VARA registry can be merged with VA pharmacy dispensing data. And the editors and reviewers understood the significance of that, which was great. So we now have a registry that looks like this. We have registry data, which is pretty simple things that are now abstracted automatically from clinical notes. We have the biorepository that I mentioned, and we now have these really, really valuable linkages with administrative data. So if with every single data element that's collected using the VA electronic health record, we have access to that in a data structure that's often referred to as the corporate data warehouse, or CDW, in the VA. We have linkages with other data sets as well that the VA sponsors. So we have linkages, for instance, with the VA Central Cancer Registry. Certain cancers are overrepresented in rheumatoid arthritis, and this puts us in a position to examine that. We have linkages with the National Death Index, which allows us to look at cause-specific mortality. We have linkages potentially with Medicare for those patients who get dual care both in the VA and outside the VA. All of this data gets loaded into something called the VA Informatics and Computing Infrastructure, or VINCI, V-I-N-C-I, and VINCI is basically a secure framework that allows us data security and allows us to do our work in a highly secure environment. So these efforts have allowed us not only to work with VARA data that's shown using a geographic heat map on the right, but also has allowed us to look at national VA data. So everybody in the VA who has rheumatoid arthritis, and I'll just point to this map on the right, basically shows, using a heat map, where all of our VARA patients come from. And one of the things you'll notice is that we cover a wide geographic area well beyond our sites, which speaks to the catchment area you see at VAs like the Denver VA and the Salt Lake City VA that have veterans that literally come hundreds of miles to get care. And so we have veterans in the registry from all over. You can see from the national map shown on the left, we have essentially veterans in every part of the United States, save Yellowstone Park, so a nice negative control there. And as of August of 2020, our last in-person, or actually virtual VARA meeting, we had over 3,000 veterans enrolled into the registry. So I want to talk a little bit more about some other research that we've done and collaborations that we have fostered. So this is Josh Baker, who is the site PI at the Philadelphia VA. And Josh joined our efforts in 2012. So we were 10 years into our effort when Josh joined. And he already has generated, this number is now about 30 VARA publications with many, many different co-authors. He has gotten a VA merit based largely on the availability of VARA. This is the VA equivalent to an R01. And he serves now on our VARA executive committee. And Josh was well aware of data in other diseases that had suggested there's this obesity paradox in diseases like diabetes, but also in rheumatoid arthritis. And we noted that early on. That's that data I showed you, that it's really underweight people who seem to be at the highest risk for RA mortality. And what Josh showed is that really that's not necessarily the whole story. And what Josh showed is that you really have to look left of registry enrollment to understand these figures. And that is, Josh looked at patients' maximum weight, so the heaviest they ever were. And this, again, was taking advantage of national data polls. And he looked back before enrollment and found their maximum weight, and then factored in their current weight. And what you see when you look at a somewhat complicated figure, but the people who are really at the highest risk for all-cause mortality were those individuals who were at some point obese and had lost weight. So it's really weight loss that marks these individuals as high risk. And this may reflect severe rheumatoid arthritis. As we know, severe rheumatoid arthritis can result in this almost cancer-like cachexia or rheumatoid cachexia that people talk about that may be mediated by some of our pro-inflammatory cytokines. Josh has taken his work well beyond that. And so Josh has asked questions about the interplay of inflammation with some of the comorbid conditions that plague rheumatoid arthritis. And this is a paper that Josh recently published in the top journal, our top rheumatology journal, Annals of Rheumatic Disease, and showed that inflammation measured with a multiplex cytokine levels or more conventional measures of disease activity predicted who would develop diabetes over follow-up, suggesting that the inflammation from rheumatoid arthritis has a direct and adverse effect in terms of glucose metabolism and your risk of developing diabetes. And in that study, he looked at multiple different cytokines that we were able to measure on those baseline samples. And you can see there's multiple cytokines, and I won't go over all these different cytokines, but there were multiple analytes that jumped out as being associated with your risk of developing diabetes. So this has led to, and I think many of you heard a talk from Dr. Brian England a few months back on his experience as a scholar in our network. Brian was a CTR scholar and recently graduated with his receipt of VA Career Development Award. But Brian's early work looked at cause-specific mortality and baseline disease activity in our cohort. And again, like Dr. Baker, Bryant was able to capitalize on the full package that makes up the VARA registry. So registry data, biobank data, and national data links. And this was a very clever study where Bryant showed that all-cause mortality was increased in rheumatoid arthritis, but not only all-cause mortality, but mortality related to cardiovascular disease, cancer, and respiratory disease. And a couple of very interesting findings out of this work. Bryant showed that your risk for mortality is directly related to your disease activity, your rheumatoid arthritis disease activity, in a dose-dependent fashion. And he also showed that the most overrepresented cause of mortality was not cardiovascular disease, like many of us would have guessed, but really lung disease is by far the most overrepresented cause of death in these men with rheumatoid arthritis. And this has been shown by other cohorts and cohorts that have included only women. And so suggesting that there's something inherently linked between rheumatoid arthritis, articular disease, and lung disease that can complicate the course for these patients. And so, our experiences with Josh Baker and Dr. England really got many of us, the older folks in the network, thinking more about how we could really use VARA as a mentoring network. And I think I don't have to convince the CTR crowd how important multiple mentors with different expertise are to the development of a trainee. And so, that's a model we have certainly embraced in the professional development core, and I think a model that many institutions and Mentee centers around the country have embraced. And so we took that really to heart in the VARA registry to build a network that would support mentoring. And so this is an example. This is just one little spoke in the wheel, but I had mentored Dr. Baker on his early career development application and his merit application I talked about. He was interested in body habitus and its interplay in rheumatoid arthritis and long-term outcomes. Bryant had done early work with cause-specific mortality, so it made perfect sense that we merge those efforts and that Bryant could learn from the expertise of Dr. Baker, which was rapidly developing at the time, and take advantage of this, the data that was mutually available. And so Bryant looked at body mass index and its relationship in terms of weight loss, the long-term changes in weight with cause-specific mortality in rheumatoid arthritis, and published this very nice paper that appeared in Arthritis Care and Research a few years ago. So Bryant now has been able to leverage VARA in many real and concrete ways, and this number is just rapidly growing. I can't keep up with it. Before we got on, Howard mentioned a paper that Bryant recently published looking at COVID-19 outcomes in the national group of patients with rheumatoid arthritis. Bryant, along with Dr. Baker, has now become a lead person in our VARA data analysis group, or DAG group, and he's now on the VARA executive committee. So in just a few short years has become just an absolutely central figure in our effort. And so this has been fantastic to watch. So Dr. England is also, so at the time he joined the VARA registry, was also doing PhD work and applying for other career development awards. And he was able to actually garner one of our CTR Scholar Awards with a project called Multi-Morbidity Patterns in Rheumatoid Arthritis. And he was able to take advantage of many of the resources of the Scholar program. And so for those of you on who aren't familiar with the Scholar program, I'd really ask you to go to our site, and we have details about this. But essentially, this is an internal K program that provides FTE support, coursework, grant writing workshops. Bryant was able to leverage expertise available at many of the methodologic workshops offered. He developed UNO collaboration directly out of the CTR. He participated in a research studio that helped hone and fine-tune his career development award application that was subsequently funded. And he participated in monthly symposium. Here's Bryant at his PhD graduation in December of 2019. He succeeded despite these mentors on either side. So that's something to be said for that. So what we've been able to do with the VARA network is, and what Bryant really has, and this as a case example, has been able to do, is that Bryant started off with this, early mentoring group that with early work that with Dr. Caleb Mishu. By 2017, it expanded that. That includes myself and others. You can see Dr. Baker. You can see Dr. Fong Yu, who was the director of our Bird Corps. And then by 2020, this has exploded really to include numerous researchers around the country, including some of my collaborators. And for those of you who haven't looked at Research Nebraska, kind of a cool tool in Research Nebraska that allows you to draw out these sort of collaborator networks. And so we did that for Bryant. And here's Bryant's network in 2015. It's a pretty small network. It includes me. And I think Caleb maybe was in there somewhere. And then by 2017, that network had grown, and this largely reflects VARA investigators. And it was about this time that Bryant interviewed and received the CTR Scholar Award. And I think by leveraging the resources in CTR and by having protected time and learning new skills, you can see what's happened to his research network just over a short period of time, 5 years, how this has grown. And so that's, impressive, really. And so he has been able to do some really, I think, groundbreaking work. I mean, that sounds like an overstatement, but he's really been able to do some very important, well-cited work in rheumatology, and he in short time has shown that respiratory disease is the most overrepresented cause of death in rheumatoid arthritis. He's published work that methotrexate is likely safe in the context of ILD, and this is important because people have steered away from this medicine in these patients, probably not justifiably. We have shown a novel biomarker for predicting who has rheumatoid arthritis lung disease. He has developed a claims-based algorithm that allows people to do big data research on this problem. I mean, that is really a important methodologic work to be able to have. He's shown that baseline pulmonary function predicts mortality in these patients. And he now has work that he's going to present soon showing disease activity in rheumatoid arthritis predicts these outcomes. And that autoantibodies may also predict outcomes. So as we have built this network, and again, this is just one part of this larger network, Dr. England and Dr. Baker's mentoring work has passed on to others. And so I have a number of names, some of whom you'll recognize as local trainees, who these two have helped to mentor. And this has included other investigators around the country. So Katie Weisham is a junior investigator who just submitted a career development award at the Puget Sound VA, Beth Wallace at the University of Michigan in Ann Arbor VA, who also has recently submitted a K-23 and a CDA award, and Namrata Singh, who has a scientist development award from the Rheumatology Research Foundation, and others. So this is really just a partial list of trainees who have benefited from this interdisciplinary collaborative mentoring network. So just a little bit more on how this has been passed on. So Dr. England has really become the primary mentor for Dr. Tate Johnson, who's one of our first-year fellows. And Tate has been very interested in looking at cardiovascular disease in rheumatoid arthritis and has done work looking at whether methotrexate, which is one of our foundational drugs to treat rheumatoid arthritis, whether it is associated with outcomes in patients, rheumatoid arthritis patients, over time. And so the hypothesis was that methotrexate would reduce mortality from cardiovascular disease, and this would largely be done through its effect, its anti-inflammatory effect. And so his study involved over 2,000 of our VARA enrollees. It was a cohort study that was done over a 10-year follow-up period. We were able to use national data to define these treatment exposures. And then Tate did a tremendous amount of work going into medical records to validate many of these cardiovascular disease outcomes, and used really state-of-the-art modeling techniques that Dr. England has mastered as part of his PhD training and really postdoctoral training, this very novel approach to look at mediation. And so what Tate showed in this work is that methotrexate indeed was protective of overall composite cardiovascular disease, but it really seemed to be particularly protective against the development of heart failure. This is an outcome we defined as hospitalization due to heart failure. And interestingly, and somewhat, I think, contradictory to what our original hypothesis was, that effect of methotrexate appeared to really be more of a direct effect. It was not an indirect effect through anti-inflammatory properties. It was really a direct effect of the medicine, which we have yet to really tease apart. And in some ways, that's very good for a career development award. So Tate is currently applying for his next career development award, and will be applying to be a participant in the UNMC Mentored Scholar Program for the coming academic year. So this has been really fun to watch, really what I refer to as the next generation in VARA for their research efforts. And so I'd also point out Tate's work recently got accepted into Annals of Rheumatic Disease, again, our top journal in rheumatology. And this speaks to Tate's efforts, but really to Brian England's efforts as he has really leveraged and mastered the resource that is VARA. So from a reviewer perspective, we hear a lot of recurrent themes when we get critiqued. And so clearly this is a platform for interdisciplinary and mentored clinical translational research focused on veterans with rheumatoid arthritis. The questions we get from reviewers often focus around the question of generalizability. You have all these men. What does that mean? And, I just always point out that, again, these are men, but the men comprise 1/3 of rheumatoid arthritis patients. In many ways appear to have more risk factors for poor outcomes. And so there is an issue of left censoring with our cohort. We enroll patients who are all comers, but one of the interesting things is, our cohort has become large enough that we have 700 to 800 individuals with incident disease, new onset disease as enrollees into the cohort. We have not enrolled in a parallel fashion control patients or healthy controls to use in many of these analyses, which is a limitation, and that's something we have heard about. So I want to end there and maybe take some questions in the chat, or there's not too many people on, I can take those directly, but hopefully what I have gotten across is how a network like this can be just invaluable, really, in mentoring and training individuals, and how you can use this sort of collaborative network really to plug trainees into multiple different types of expertise in a very efficient way. And I think I'll end there and end with this thanks. And this is the network that we've developed across the United States of our VARA site PIs. Really just a fantastic group to work with. You'll recognize some of them, but we have new sites in Boston. We have a new site in, I mentioned, Ann Arbor, Michigan, Augusta, Georgia. We have a new site in Durham, North Carolina. And we continue to grow, and we continue to get questions about participating in this. Again, which kind of amazes me given our lack of funding that we provide these sites. So I'll end there. Thanks for having me.
Ted, if I can start off the questioning, as I'm not shy. First of all, you skipped the critique that we don't believe you're so successful. We actually got that. And I mean, just wonderful. And wonderful, science as well as mentoring and bringing new people in science. So, how to translate this to other fields. And certainly, I think as you pointed out, the VA, is a wonderful resource, right? The first health record, and, once you're through all the regulatory hoops, the ability to do all this. Have you been approached by specialists in other fields to set up a similar study? Or have any of your trainees gotten interested in a different disease and tried to do that? So I'm just curious on if you're really successful, you'll have more investigators and questions. You're not there yet.
No, it's a great question. And I would, I was remiss, I think, early on to say that with our renewal of the CTR, and I want everyone to know that the VA is one of our partners on the renewal, the Omaha VA, and there are VA facilities not only in Omaha but in Lincoln, Grand Island, across our state and across the Midwest and across the country. And so, plugging into the VA sounds a lot harder than it is, I think, and not that it doesn't come without challenges, it does. But to your point, though, we have had spinoffs, so in rheumatology, there are two other registries that were developed that used VARA as a direct model. There's actually one that's led by Laron Kaplan and Andreas Reimold that looks at spondyloarthropathy. It's called the Pulsar Registry. And there's one that's very cleverly called Crystal that looks at patients with gout. So there are these two spinoffs. We have been approached by people interested in other conditions as well. I spoke with VA investigators from another site who were interested in seizure disorders, actually, and particularly seizure disorders related to traumatic brain injury.
Right. Yeah.
But I don't know that all of them have had the same success, and I don't know the exact reasons for that, but I can say that luck plays into things, and I think personal relationships can't be underemphasized. So when we developed this early cohort of people interested, these were people I had already had a relationship with, or collaborators had a relationship with. So there was this kind of natural, there was this natural trust that was there immediately that really helps you build something like this. So, so I think there is some luck and some forethought that has to go in with who you invite into these efforts early on.
Thanks. And I think we can't, the personal relationship, the trust, right? I mean, science and medicine and all that is based on that. So, it's difficult. Some people start off, oh, build a network and write to all these people, and then it never gels, and it's, hopefully we'll get back to meetings where we can see each other and build upon this.
I agree. We have a national meeting each year, which is, I think, most of us think has really been integral to kind of keeping this effort going. And so we did it last year virtually. It was okay. And we have been forced for 2021 to go virtually again, even though it's not until this fall. But we had to pull the trigger on our planning. And so we're going virtually, and that's a disappointment.
Yeah.
But
and it's harder for new investigators, I think.
Absolutely.
Much harder, because to speak up in a Zoom versus, people seeing you and things, I think.
Absolutely. So we, I will say that, we have sort of one-offs with new investigators and new sites. And so that really helps. And so we really try to foster these relationships. And one of the things we try to do is really strategically select sites that bring something new to the table. They have maybe it's a new methodology, a new interest. There's many outcomes that interest people in rheumatoid arthritis. And so, if you get 7 sites who are all interested in cardiovascular disease outcomes, that can create, problems. And so we really try to pick sites who have complementary, interests. Well, again, want to thank everyone for being on today. I don't want to hold people on in silence. So, want to thank people, and I look forward to working with the VA even more directly when we get the CTR renewed. So
think positive.
Think positive. All right. Thanks, everyone.
Thanks, Ted.